Hello everyone, good afternoon. Thank you so much for tuning in. I'm going to be going through the notes that are going to be on the exam. This is going to be part 2 of the microbe diversity, the ones that we started last time that the professor went over. This is the second half of it. Alright, so when we're talking about finding the microbes in natural environments, we would need ways to identify them. So this is kind of a review where we could use rRNA identification. So first you're going to amplify the 16S rRNA using PCR, and again you're going to probably be looking for an unknown organism. Then you're going to determine the phylogenetic relationship to the 16S rDNA sequences of the known genera prokaryotes. You're going to be using databases such as BLAST. That's kind of the common one. Another important way to use PCR and cell detection, I'm just going down the slides to have this icon. The next one we'll be talking about the primer design. So now we're down here. So we talked about probes here, whole genome shotgun. Those are also important ways. More on those in other notes, but primer design is the next one that is specifically highlighted. So in cell detection and primer design for PCR, you're going to be using the database of 16S rDNA sequences to guide the PCR nucleotide primer design. You could have universal primers, and these are going to be chosen for a part that's complementary, and it will be 100% conserved in all 16S rRDNA. So a universal one, that's just going to get what's going to be conserved throughout all species. Again, primers could also be unique to both archaea and bacteria, or just archaea. The primers could also be labeled with a fluorophone, and this could be used as a probe for a target sequence, and you're going to use an optically detectable method in order to find that certain sequence of interest. Again, with our primers, we have a forward and a reverse primer, so we're going to need to know the sequence and how many base pairs over for that region of the DNA that we want to PCR. Now let's go into the environmental shotgun sequencing. This is when we're going to take the whole genome and then we're going to cut it by restriction enzymes. So I have them listed here as restrictases just to save some time. Fragments are then cloned into a sequencing vector, aka a plasmid. That's going to be our vector here, a plasmid, a designed plasmid. The plasmid clones are then sequenced. And again, it's called a shotgun method because all these random bits of the whole genome of whatever we're trying to sequence, they're all going to be random pieces. So we're going to have all these different DNA sequences at the ends of these plasmid clones. You could use this as a tool to explore the genomic complexity of microbial populations. Bacteria will have different pieces of DNA. Again, the shotgun method, talking about how all those fragments, after they're cloned, they're just going to make different sequences, and then you try to order the sequences all together in the end product. We could also be using ribotyping, so this is going to be in natural environments. First, you're going to have 3 different strains of bacteria, then you extract and cut the DNA of those said strains using, once again, a restriction enzyme. You're going to then do gel electrophoresis to separate the fragments by size. And afterwards, you could use a probe based on the 16S rDNA. Let me see where I am at. Yep, okay. So then you could reveal different prokaryotes widely separated from one another on a 16S rDNA tree. We're going to be looking at the ribotype profile when we are finished doing the electrophoresis and then the nylon filter, and then you're going to also hybridize it. Once we're finished with all those steps, we're going to be looking at this banding pattern or the barcode for the particular 16S rDNA sequence. Okay, so cultivating prokaryotes. So used to be believed that a majority of the free-living organisms were uncultivatable. Sorry about that. And the reasons given would be they didn't know, there was impatience because of their growing time was too slow, or there wasn't just any knowledge about or imagination about the chemistry or the moisture, the nutrients needed to grow certain organisms. All right, our next marked slide will be about different taxonomically useful and diverse bacteria. So we're just going to be listing the species now. Actually, do you know what? I think I'm going to write all these out so I better remember them. I do have them all written out here already, but I think it's best if we get back into the habit of trying to remember some of these. All right, so our first species is Deinococcus. Yes, so the pen clicks are going to return here, but Deinococcus is highly resistant to radiation. It's a chemoheterotroph, and it's found in soil, ground meat, and dust. So I'm just going to say soil and chemohet. All right, that's the first species here. Our next species is going to be Thermus. This is the Thermus aquaticus. It's a species of Thermus. This is going to be very familiar because this hot spring bacteria is where we get the Taq polymerase from. And of course, that's for PCR. They are thermophiles. They really like optimum temperatures of about 70 degrees to 72 degrees. Very specialized for hot water tanks. Let's see. The enzyme of the Taq polymerase, that enzyme, so polymerase, it's been cloned and expressed in E. coli. And this is going to be useful, again, in PCR reactions. So that's how the market for Taq polymerase is supplied. They're actually expressing it in E. coli instead of looking for the Thermus aquaticus itself. All right, now we're going to be talking about the proteobacteria. These are going to be gamma proteobacteria. Proteobacteria are going to be gram-negative. And there could be pathogenic species in here as well. So the first one would be the Pseudomonas, or the fluorescent pseudomonids. Okay, so we've actually talked about these Pseudomonas before. There are a few species, aeruginosa, fluorescens, and syringae. Okay, so there's something important about the syringae one. Let me just get back up here for a little bit. No, I guess. Or mine, I was remembering something else. Okay, so Pseudomonas are going to have these fluorescent compounds that are yellowish green. And they can't ferment due to them being obligate Arabs. We could use a wide range of organic compounds as energy sources. Applications of Pseudomonas. So they could degrade. These are pretty cool. They could degrade camphor, toluene octane. Okay, here's where syringae comes in. So we see all these purposes. They're kind of like contamination control of like octane. But P. syringae, this one is specifically listed here. The outer membrane protein complex will nucleate ice crystals. And this causes frost just on the leaves. And the bacteria will use this to invade the plant. And then you also have ice minus strains. And then when you add the ice minus strain, this will actually repair the plant. This is an example of a bioengineered organism. All right, the next gamma proteobacteria would be the E. coli. This is another famous one. E. coli, again, are bacilli. They're going to be in the intestines. They're the most extensively studied organism. They're going to be also members of the Enterobacteriaceae. They generate ATP by two methods, I think. Yeah, we have oxidative towards the organic compounds. And then we could also use fermentation under, of course, anaerobic conditions. We could use E. coli. The big deal with E. coli is that you could use them to produce proteins using recombinant DNA technology. Some proteins that have been used for this would be the insulin, as well as growth hormones. And the plasmids are going to be amplified by E. coli and the COVID-19 vaccine. So an interesting nod to the pandemic that happened, that's still happening in some areas actually, fortunately. Yeah, E. coli played a big role in creating those vaccines and all the ingredients in them. All right, more proteobacteria. We're going to go to Xanthomonas. This is the genus that makes the xanthan gum. They're going to secrete that polysaccharide onto the medium. And the specific species is Campestris. This is going to be making that xanthan gum for food, the food industry, as well as oil recovery. We also have genus Acidithiobacillus. That is an interesting genus name. These are going to be acidophiles, hence the name acido. Spelt it wrong. Sorry about that. There we go. They like a pH less than 4. They're going to oxidize sulfur or iron 2 as the energy source. And ferrooxidans, which is a species from Acidithiobacillus. Thio again means sulfur, so there's another hint there. This is going to grow on Fe2+, as the substrate. We're going to be anaerobic sometimes, so in anaerobic conditions, this bacteria, the ferrooxidans, will be using the reduced forms of sulfur as the electron acceptor. Very interesting stuff. All right, now we are going into the alpha proteobacteria. There we go. All right, alpha proteobacteria include the Rhizonium, Zobium, I should say. These are going to be bacilli. They're going to be aerobic chemoheterotrophs. They're going to be in the soil. They're going to be in root hairs of the linguine plants. They're going to fix the nitrogen for the plants. And when they're fixing nitrogens to benefit the plant, they're going to convert about 200 million tons of nitrogen to ammonia, or an NH3. We also have Agrobacterium. This is another genus of proteobacteria in the alpha division. These are going to be flagellated. And rods again. This is going to be the same here. They're both aerobic chemoheterotrophs, so we could actually just copy this over. Whoops. They're also found in the soil. They're going to carry this very large and famous plasmid called the TI plasmid. Again, this is one of those species that's used in like bioengineering and biotech. Okay, so the TI plasmid, this is going to encode functions for the transfer of the plasmid DNA or the tDNA into plant cells. Once that happens, again in biotech, we're going to be using this to transfer foreign genes into crop plants. So something like what Monsanto does, they would take this TI plasmid and we're going to stably transfer, safely, I should say. So that's like an example. So when we're using the TI plasmid, we're going to be able to allow for the stable transfer of foreign genes into crops. These could be like growth hormones, etc., but that's how we're going to be able to do some biotech with plants as well as bacteria. We have the TI plasmid of Agrobacterium, and we're going to allow it to transfer the tDNA into the plant cell. More alpha division, we're going to go to Zimomonas. These are going to be found in the sugar-rich plant extracts such as palm, wine, sugarcane, and cider. They use fermentation or respiration. They're going to be using the Entner DOUDOROF pathway with ethanol as their fermentation. So I'm just going to probably underline that. They're going to be better than yeast in large-scale ethanol production. Again, for that ethanol production, you need to think about profit here and cost effectiveness. All right, the next genus is Gluconobacter. These are going to be ellipsoidal or rod-shaped. They're going to be obligate aerobic chemoheterotrophs, so they need oxygen. They're going to oxidize ethanol into acetic acid. But it stops here. Doesn't go any further. We're going to be using Gluconobacter for vinegar production. Again, acetic acid is a component of vinegar. Oh, I just realized something. Some of these have the little icon, some of them do not. So, this slide of Xenomomus and Gluconobacter, these aren't like marked in the slide, so that's just like extra information, you know. Firmicutes also aren't marked. So firmicutes, I'm just going to go up here where I wrote it down, thinking that they were going to be on the test, which is up here somewhere. Firmicutes are gram-positive. There's going to be one genus here. They have low G and C content. Clostridium is rod-shaped, usually flagellated. They're circularly anaerobic. They make those endospores like Clostridium botulinum. They have been used also to produce acetone in World War I through mixed fermentation. So that's just something on the side there. Firmicutes won't be on the test according to the notes because they weren't marked. This firmicute, however, will be Lactobacillus, Staphylococcus, and Bacillus, the cluster here. So Lactobacillus, Staphylococcus, not Streptococcus, and Bacillus. They could grow in the absence or presence of oxygen, making them all facultative. We could ferment simple sugars into some products here. We have lactic, which is going to be Lactobacillus and Pediococcus. Or we could have lactic, ethanol, and CO2 when we're talking about the Leuconostoc or the heterofermentive species, Lactobacillus. I should also put Lactobacillus here too. So this is a fermentative Lactobacillus for only lactic acid. However, if we could make lactic acid, ethanol, and CO2, that's going to be Leuconostoc and the heterofermentative Lactobacillus species. The shapes are going to be Lactobacillus, of course, bacillus equals rod. Leuconostoc and Pediococcus are cocci. They're going to make cheeses, fermented milk products, alongside the strains of Streptococcus. Streptococcus, our next one. I'm going to just write strept. These are going to be, of course, spherical because coccus means like grape or spherical. Generate ATP by converting glucose into 2 molecules of lactic. Cremoris is going to be used for hard pressed and soft ripened cheeses. This is a species, also known as Lactococcus lactis subspecies Cremoris. That's the new name of it. This is going to be, again, hard cheese like cheddar, or soft cheese such as camembert. The lactobacillus as well as the streptococci account for world output of dairy products. This is going to be 20 million metric tons per year. It's about 50 billion. Okay, next one would be Staphylococcus. We skipped over Bacillus. There isn't a mark there, so we're just going to skip over it. We're still in the Firmicutes. We're going to be talking about staph now. Remember, strep, here's a different thing between staph and strep. So I accidentally said this earlier, but Streptococcus are going to be in chains. Staph means grapes. Sorry about that. I got mixed up there. My brain ain't working. Staph is like grapes. Strep is going to be like a line. Staph are going to be using sugars as their main source of energy. They could use both fermentation and respiratory means of metabolism. They're going to be found on the skin. They could tolerate salty environments, so they're able to grow there. There's a virulence factor called protein A in aureus. This virulence factor, it prevents IgG, or one of the types of antibodies, from stimulating the immune system. And how it does this, it's going to bind to the Fc region, or the constant region of this antibody, this immunoglobulin. I want to highlight protein A and maybe IgG in the Fc region, because that's what it stops. We could use protein A in purification and analytical procedures. One of these situations would be ELISA, where if an antibody is available to a molecule that needs to be isolated, we could actually have whole cell preparations of Staph aureus or the protein A could be used to bind to the antibody and the target molecule as a complex. Again, some throwback to cell bio with ELISA there. Acintobacteria, those aren't marked. Okay, fungi, these are actually going to be marked and on the test, so I'm going to write them down here from the notes. Fungi, eukaryotic, they make spores, bisexual and asexual. Hyphae, that means apical growth. I'm going to write that down. And then you also have yeasts, which just bud. They're heterotrophic. They're going to absorb nutrients through their membranes. They secrete enzymes to use high molecular nutrients. We're going to have cell walls of chitin in fungi, which is kind of a review from every bio class ever. Alrighty, next we have two types here. We have our first type, the Diomycota. This type of fungi These are going to be diverse. They're usually aquatic, which is very interesting. Rhizophyticus rosea. Rhizophyticus rosea. This is going to be able to decompose cellulose. Rosea kind of reminds me, oh, that's a rose. Plants have cellulose, so that one is able to degrade cellulose. That's how I'm probably going to remember it, hopefully. Our next one is Glomeromycota. These fungi are going to be arbuscular mycorrhizal fungi. Or arbuscular, yeah. Yeah, I'm almost finished. They're symbiotic and asexual. Mycorrhiza, this is going to be a reference to their association with mycelium and the root of a plant working together. The mycelium gets the nutrients. Most important is phosphate, and it's going to give that to the plant. The plant gives the sugars to the mycelium. So it's kind of this very nice cycle here. We still have a few more types of fungi. We have Zygomycota. These are going to have chitosan in their cell wall. We're going to have the saprophytes mucor and Rhizopus. Rhizopus nigricans. Will be used for citric acid. We were just talking about that actually earlier. The earlier video. Okay, Ascomycota. This is the largest division. We're going to have yeasts or segmented filaments. We're going to have chitin and glucans. Such as mannan, or that's another part of their cell wall in yeast. There are two organisms. However, sexual reproduction leads to the formation of spores. In the ascus, we have Neurospora or bread mold, or Saccharomyces, which is brewer's or baker's yeast. Basidiomycota and the Deuteromycetes, so these are the last two groups. They're going to make the sexual spores in the Basidium. Their vegetative structures either yeast or separate mycelium. Remember here we had filamentous in the Ascomycota. Alright, back to Basidiomycota. We are going to have glucans and chitin again. The agaricus species or mushrooms are cultivated for human consumption. So agaricus mushrooms. It's a main player right there. Okay, last group are the Deuteromycetes. These are going to be in their asexual stage only. We're going to have separate mycelium or yeast, just like the Bacillium mycota. So I might just copy that over like this. See how well that copied over. All right. The cell wall again would be glucan and chitin. We're going to have aspergillus, and then different types of aspergillus. The first one is Niger. This one is going to be citric and gluconic acid. That's a product. We also have oryzae. That looks like rice, oryzae, you know. Kind of reminds me of the word rice. And guess what they're going to be using? They're going to be fermentation of rice and soya. So like soy sauce, I'm guessing. No, I need to do more research on that. Soya product, so one of them that comes into mind is of course soy sauce, and then you also have penicillin notatum. This is the first producer of penicillin. And remember, the penicillin producer is a different strain, but the first time it was found, it was this penicillin notatum. Let me look quickly. Yeah, now they use penicillin chrysogenum. So the first time was notatum, but now they use chrysogenum, I think. Alrighty, those are all the different diverse microorganisms that will be covered according to the professor for the test. Thank you so much for tuning in. Please have a good rest of the day and do something nice for someone. Take care.